think we're about ready to start the last session of the day. I know you've had a long day and it'll be a great day. My name is Mary Riley. I'm a neurologist in Queen Square in London, and I'm delighted to be here both on my own behalf, but also I'm a past president of the PNS and I'm going to be the chair of the CMTR. So I think this is a great initiative and we're very much looking forward to collaborating. We've already come up with a list of jobs that you don't know about yet, which we might talk about later. So what we're going to do first of all this afternoon is have a summary from each of the subgroups so that people that weren't at those sessions can understand what was discussed. Then Pavel is going to give his perspective on putting it all together, and then we'll open it up to a general discussion. And I think it's important while we're doing this to remember what was said at the beginning, what we're all about. What this is, you know, looking to see can we all work better together. Is there an appetite for having some, you know, European CMT, although we've told you now it has to be worldwide CMT association. And I think there's some very specific questions coming out today that maybe only patients can answer. So the data, you know, sharing keeps coming up. And my personal belief has been for many years that there should be no commercial or hospital or academic-based lab doing genetic testing that hasn't got a compulsory, they have to upload the information that they wouldn't get licensed. Now, we can't make that happen, we've all tried, but I suspect patient organisations, the European one, could actually politically make that happen because it's a big thing to make happen, but I think so. So having said that, we'll start this afternoon. The first session today was the Bio and Medical Sciences, and Rafael Severa is going to summarise that for us. Rafael, thank you.
Hello, good afternoon. The working group that I'm going to summarize was the one on biomedical sciences and genetics, and the object of this work group was to try to find out or to try to delineate the current roadblocks and challenges in basic and translational research, and also to have a series of proposals to see how we can solve them together. So the first of our talks was by Davide Pareison, and it versed on the way in which we try to focus the information, the data collection. The data collection has to go from gene mutations to treatment, and we don't have to think of data collection only as the clinical perspective or the biological perspective, but a more global data collection. And also that we need to be together against CMT, centered on the patient, but also in many other ways, and also the industry, all of the different stakeholders have to take charge of the situation and try to go together to solve the clinical problem. The main hurdles that Davide talked about was basically the funding. The funding, comparing it also with other diseases, even with other rare diseases like ALS, we are less funded than they are, and to other more frequent diseases. So it's funding is really one of the main concerns and the main hurdles that we face. Also, the heterogeneity of CMT, the need for good disease models, because it's not the same. Different type of CMT have different mechanisms, different disease models will be necessary, and the way to go from the disease models to the actual patient is something that is very challenging, as is the data collection and the management of the data, and also the challenges in clinical trials. In clinical trials, mainly the slow progression of the disease and the variability between different types of disease and even between different families, and also the shortage of personnel. Not all our hurdles, he also presented a series of collaborative initiatives that are already taking place. And he presented examples of collaboration, of sharing of genetic data, for example, in the international, the INC, and also sharing for having more information about rare forms of CMT, for example, CMT4B, and the way in which international collaborations have managed to give us more clinical information and more robust information about these diseases. And he also gave us the example about the Italian National Network and how the registry gave also more local information about mutational distribution and about certain patient-reported outcomes that are very important to us. And the proposals that were shared in the discussion was that to try to impulse the data sharing, the genetic and the clinical and the natural history, the creation of an infrastructure for clinical trial conduction that may be similar in different centers, but have to be centered, and the preparation for gene therapy, the further connection of stakeholders, not only stakeholders from the clinical perspective, but also stakeholders from pharma, stakeholders from patient associations. It's very important for us to all go in the same direction. And the importance of registries, which also is something that the data sharing and the data collection is something that needs to be available and needs to be shared in different ways. The main concerns in the discussion in this talk were the challenges in gene therapy, the awareness of the disease, that is sometimes we feel that it's not the population in general is not so aware of the disease as, for example, of other diseases, which are maybe even less frequent, and the lack of funding. And there was also a concern about the number of different organizations and the difference to go and the importance of going together in the same direction. And that was the first talk. The second talk was done by Francesc Palau, and it versed on the homeostatic vision for the improvement and clinical and therapeutics in CMT. So what this vision is based on is in the complex biology of the genes and of the complex biology that is underlying CMT, and on the need to take that to the patient. There's a very numerous, there's a large number of genes, a large number of structural proteins, and we have to try to define the pathophysiology so that we can try to aim for different treatments. So we have to move from reactive medicine to a more proactive, a more precision-based medicine. Dr. Palau, he proposed a multi-omics approach, which you can see here, in which the individual, the shared genome, and the societies in the middle of all of the data that is collected, data about genetics, about genetic counseling, about clinical in different situations, and that it has to be translated to the scientific through the scientific path, through rare diseases programs, and then trying to approach the disease by different ways, by trying to find the biomarkers, the therapeutic molecular targets, the different regulatory and epigenetic profiles, etc. An example of this that was provided was the work in GDAP1, in which the work went from a clinical observation to a molecular, to more the discovery of a gene and the development of a mouse model, and now the development of treatments that are actually that go against some of the alterations that were found in that mouse model. And the mouse model has also to be validated clinically, and so that closes the circle with the clinical information, the biological perspective, and the translation towards the clinical pathway and the patient, which is the most important. And the main discussion versed on how can we improve the roadmap and reduce the time gap from these translational research, from these researchers in the basic lab, to actually the patient, and how can we reduce the time that it takes. And that's the difficulty sometimes in moving from mouse models or from animal models to patients. And we have to find a way to accelerate certain pathways so that we can get to patients at a shorter time that we do right now. There was also concern about the problem of the variance of undetermined significance, and also about about if we could find a way in which different labs, basic labs, could collaborate and maybe specialize in certain pathways so that we could all gain from this collaboration. And the last talk was by Wolfgang and Elena Pernis, and it was about a deep experimental and clinical profiling of CMTR patients at a large scale. And so basically what the talk was about is in trying not to stop at this level, trying not to stop at the genetic diagnosis, trying to find if the genetic diagnosis could lead to a more complete phenotyping of the patient. And we have to take into account that a large number of our patients do not have a clear genetic diagnosis. And so we have to try to see if we can phenotype them better. And they proposed a very interesting model which was based on the study of fibroblasts and a machine learning model in which they studied different variables in the fibroblasts. And they studied with a machine learning model, they tried to phenotype the different subtypes of CMT and see if this model could predict and could we could predict the different phenotypes or the different, if the fibroblasts we receive from a patient could be compatible with a certain phenotype. They also raised a certain concern about the way in we can share data, because they found that there were certain difficulties in the sharing of data between their two sites. And so the hurdles were especially the legal barriers, the transfer cost, and the loss of control of the data. And they proposed also a model based on a cloud-based shared workspace, in which there could be different types of collaboration. Maybe they could simply be accessed from different sites to the data. There could be a linkage, there could be linking of computers, but not of the actual data, or even linking certain the results of the certain models without linking the actual data. And so that's the proposal that they presented. And globally in the discussion, I think that the major challenges and the major difficulties that were addressed and that were proposed are basically the funding, the infrafinanciation that sometimes that we find, and the delivery to patients, the quick delivery and the quick delivery of basic research to the patient population, which is what all of us clinicians want.
Thank you very much. I would suggest, unless there's burning questions, we listen to the talks first and then have a general discussion, if everyone's happy with that. The next speaker is Michael Dorn, who's going to speak about the clinical diagnosis and treatment session. Thanks.
Thank you very much. I feel honored to be allowed to summarize our workshop, which was really, really interesting. So if I only had 2 words to characterize our workshop, I would say trajectories and crossroads. So following up with the road metaphor. And I think a main challenge is to combine all the knowledge and ideas and dreams and concepts that come from scientists, clinicians, and patients all in one session. So that was the aim today. And I think we all know that the main prerequisite for treatment is diagnosis. And treatment doesn't only involve cure, but also care. That was one point that was stressed, even especially in the summary. So counseling and also pre-implantation counseling is really important. And for that we need a safe diagnosis. So we need to know what we are doing. And we know that 127 genes are known today, as of today. And looking back to the beginning of the road, we know that only a little bit more than 30 years ago, no gene was known, and we are very honored that we have Vincent and Peter here, who were the first ones to describe PMP22 only in 1992, so that's not long ago, and we are moving very fast, 127 genes today. But still the diagnostic gap is massive, especially in axonal CMT, it's about 50 to 75%. So from now on, the challenge is shifting from the generation of data to the interpretation of data. So there's an overwhelming amount of data, and we can actually basically sequence a genome in less than 30 hours. That's what we learned today. But we need to interpret this. And in order to be able to do that better, we need to curate evidence and also exclude genes from panels that are not relevant, and at the same time share our data of those unsolved cases to enable new discoveries and better diagnosis. So on the way from diagnosis to treatment, we have some major challenges. And one is that actually so many different cell types are involved in CMT. Motor and sensory neurons, and also Schwann cells. And then within the cells, we have different compartments that are being involved. So, for instance, the cytoskeleton, mitochondria, ER and Golgi. And then we have different mechanisms that are being involved. I'm just naming some, which are axonal transport, synaptic transmission, intracellular trafficking, and so on. So it's very difficult from this to develop therapies. There are some approaches that try to encompass all those kinds of mechanisms, for instance, inhibitors of SAM1 or HDAC6, which we have discussed about, but most of the therapeutic approaches actually are very pathomechanism specific. And we know that treatment has, of course, the aim to be efficient, safe, durable, and predictable. And then we have challenges like genetic heterogeneity, variable expression, the need for larger models. Let's just remind ourselves that these diseases are length dependent, so we need long axons in animal models, and also age dependent, so models that live long enough to be representative. And then we have problems with toxicity and immunogenicity. And let's also take under consideration that loss of function and gain of function mechanisms require different approaches, and that also the routes of delivery are complicated and depend on the target organ or cell type that we are facing. So the good news is that there are some drugs or concepts in preclinical and clinical trials right now. And we discussed the different types of drugs. For instance, within the field of genetic therapy, drugs that require viral delivery, CRISPR-Cas, or RNA-I and ASOs for translation modification. And then there are small molecules that might exploit the opportunity of drug repurposing. So let me just name some preclinical examples for CMTX, CMT4C, CMT4J, CMT1B, and so on. I noted some more. And then clinical trials, for instance, on GAN, CMT2S, CMT1A, and so on. So this is really good news. There is something happening. We are moving. And then for pharmacological approaches, we have also learned a lot. For instance, there's more than 6 years of clinical trial experience on the combination of baclofen, sorbitol, and naltrexone. And we have learned a bunch of really useful things on the negative trials on ascorbic acid, for instance, that we need better biomarkers. I'm coming back to that topic a little bit later. Then there are other examples, for instance, on receptor antagonist for progesterone and glucocorticoids that have good data in animal models, but actually no pharma company was there to really, to fund this and for partnership. So this is the crossroads point that I'm going to stress. The problem is that these diseases are rare and that effect sizes are somewhat small. So pharma companies don't want to invest in that. So this is a point that we need to discuss, that we need to put pressure and to increase awareness that this is still very important, even though it's not ALS or cancer, but it's important to work on this and that we need natural history studies and that we need biomarkers for this. A positive example is SORD neuropathy. We have all heard about this earlier today. So when the gene SORD was discovered in 2020, I think nobody would have really guessed that only 2 years later, a first phase 3 trial was initiated. The enzyme sorbitol dehydrogenase is not really working. That inhibits the conversion of sorbitol to fructose. And drugs have already been investigated on for diabetes that inhibit the upstream enzyme aldose reductase. So repurposing of these types of drugs has helped to initiate a clinical trial that is already running for about a year now, which is good news. And we are optimistic, like carefully optimistic, that this might be a really good first example. But also for that, we definitely need outcome parameters. So natural history is one thing, and we discussed the topic of biomarkers in this workshop. There are diagnostic biomarkers that help us to identify patients, but then also prognostic and predictive biomarkers that help us measure if treatment is efficient and how a disease progresses. A good biomarker should, of course, be relevant, reliable, sensitive, specific, practical, and cheap. So this is not happening, right? Yeah. So we discussed the example of sural nerve biopsies, for instance. They are great. They are giving us lots of information, but you cannot repeat them. We only have 2 sural nerves per patient, so it's not so practical. And then we have things that are just too expensive. Within the topic of biomarkers, we discussed the clinical biomarkers, for instance, CMT-NS and CMT-ES, and we talked about a rush analysis that has been done for the CMT-ES score to make the score more representative. And we also discussed biological biomarkers. For instance, I remember that we had a very good discussion on NFL, which is an unspecific marker of axonal loss. And we had a good question, which is, can it still reflect the disease dynamics when many axons have already been lost? Because it's a dynamic disease. And then there are other diseases like ALS that have a different background. And actually, what is the effect of comorbidities on this one? So it's important to further investigate those biomarkers. We discussed imaging biomarkers, such as MRI. For instance, you can measure cross-sectional areas there. You can measure the fat fraction, and this is actually a marker that is significantly different after 1 year in CMT1A patients. And there are also more fancy markers like magnetization transfer imaging that might be able to distinguish between axonal and demyelinating changes. And we also talked about ultrasound that is complementary as a method that, for instance, can measure muscle thickness and echogenicity, but also cross-sectional areas of nerves and how homogeneously they are enlarged. As a little bit of a perspective of this road that we're talking on, we had 2 aspects. One is translation. So we need to know whether those biomarkers actually apply equally on animal models, because how can we conclude if we don't have comparable biomarkers? And there are ongoing projects, for instance, MPZ mice that we talked about. And then the second aspect is transfer. So how do functional biomarkers reflect pathomechanisms and also ultrastructural changes? So this is a lot that still needs doing, and I think we're working on this. This is a good point. If we look back at the beginning of the road, there was nothing, and now we have a lot, but every question raises another question. So I think it's important to come together here, us, scientists, clinicians, patients, and pharma companies, to find those crossroads and further move on in our trajectory. Thank you.
Thank you, Meike. And the third speaker is Roberto Conataro, who's going to talk about the therapeutic practice and feedback.
First of all, I want to thank the organization for inviting me. And I'm involved in CMT since at least 15 years. So I'm very linked to especially the Italian organization on CMT. So I'm particularly proud to be there today. And me, I'm a nutritionist, so I'm a bit far from the first topic that I listened to the working group, but I think that there is some very important point for the presentation of Dr. Kale, and that he presented a kind of surgery they call it triple arthrodesis that has some advantage because it's less invasive than the other kind of surgery. Obviously, it's a surgery, so it cannot be not invasive. And it is a very interesting outcome in last week from recovering and rehabilitation, and he doesn't report any infection. And so this is an important point because sometimes it's possible in other kind of surgery, infection are possible happens. And other key points that I wanna point out is the timing of surgery, because it's important to not have it too soon, but not too late. In effect, he presented a case that the surgery was done after 14 years, and okay, there was a good result, but probably not so good that the surgery could be operated before than 14 years. So he reported the better time from 20 to 40 years to have the surgery. And the other presentation from Dr. Ramdari, I score it, I hope so. And I think is a very important point. Probably because I am an ex-personal trainer, so for me is I'm particularly linked to this topic. But the training on people that suffer CMT, and particularly strength training. And I think that is a very important point, because often clinicians, physiotherapists are scared to propose strength training. But probably because they don't know exactly what is strength training and what happens in our body when we train strength. And often when normal people think to strength training, look at bodybuilder, too much muscle, but no, strength training is a neuromuscular adaptation. And this is, I think, the key point of strength training, neuromuscular. When we train with strength, obviously we train our muscle, but before we train our nervous system, central and peripheral nervous system. And this is very important, because we know that unfortunately there is a degeneration, a progressive degeneration. But if we keep training the nervous system, this degeneration is slow. And I saw in people, especially in male with CMT that train with weight, that there is a better outcome. So I think that is very important. Obviously, it should be proposed in the right way. There should be a progressive load. There's not be too much, but progressive increasing the weight and the intensity. And I would like to report a case report that we submitted of a guy that is 18 with CMT 1A. And he's training with weight, and he had a very good outcome and improving in all clinical outcome assessment. And it's important because sometimes there is a loss of motivation on who train with weight. I see that people train for 1, 2, 3 months, then give up. In this guy, he's 18, so he wanna be with six-pack, with muscle, so he's very motivated. But it worked, because he had a very important improvement. It could be nice to set up a trial that could assess the importance and the protocol to train with CMT. Probably in the future, and link it to a correct nutrition, because if I train but if I don't feed my muscle, that's not so that's not okay. And last presentation is another important topic, and is the clinical outcome assessment, because we have to have tools to judge, to see what's happened on our patients. And it's not easy, because we have some tools to assess it, but often they need a lot of time, a lot of skills on who see what's happened. And so is needed a training of professional that assess the clinical outcome. The time is needed. Often there is the needed of a specific room or space to assess the trial or the outcome assessment. And there is no uniformity at the moment, because there is not one battery of assessment recognized. Dr. Mandarakas proposed a panel from childhood to adult to assess clinical outcome. And it could be very interesting. There is a platform that could be assessed. Obviously, it's in English because she's come from Australia, and it could be interesting to have a translation, for example, in European language, German, Italian, Spanish, French, of course, because if we use it, it's okay. But if you wanna propose it to our patients, it's not easy for them to access it in English. But I think that is another important topic, and in which one we should work, because it's another needs a clear, unique clinical outcome assessment where we could recognize all. And especially not only for scientific paper, where we could have more time, more space, but even in the daily practice to assess how treatment, physical, physiotherapeutic, dietetic, and so on could go, and if we are really in the right direction. And with that, I reported the very interesting report, and then we are open to all the questions for the public. Thank you.
Thank you very much, Roberto. So we're now going to ask Pavel Seaman, who's from Prague, to just have some comments on the day generally.
Okay, so thank you very much for this honor. It's probably also due to my age. I partly will comment the day, and also my personal some suggestion or memories. I'm investigating patients with CMTs for around 26 years in Prague. And I had also excellent examples when I started, like Vincent Timmerman and Peter Jonuche. I tried to follow their, it was like gurus taught to do. And I remember meetings, also my teacher in Erlangen, Professor Rautenstrauss, who is unfortunately no more here. But I remember meetings in Antwerp organized by these two excellent experts, and we were earlier in Europe than in US. Then in 2003, the US experts said we have to, they were also in Antwerp, and they organized North American Charcot-Marie-Tooth Consortium in London, Ontario. I visited their meeting, I remember, with Mike Shai. But and then after that, PNS, Peripheral Nerve Society, developed, I think, a lot of, or quite a lot in direction of hereditary neuropathies, because it's led and organized a lot of by experts who are involved in hereditary neuropathies, like Mary, or also some previous chairs. So CMT become important in this world organization, and so I think it's excellent that these two gentlemen had this dream and organized this Charcot-Marie-Tooth European Federation. When I started 1997 in Prague to test patients, 2 years later we founded Czech Charcot-Marie-Tooth Association, because I have seen in US it's done like this. So also in Czech Republic, but there was no European Federation. So this is a big step further, and I appreciate it. I think this is very important. Maybe it's said the devil is always in the detail. What we have heard here about this is few small comments. We have heard about 27 genes. I remember the time when we had no genes for axonal Charcot-Marie-Tooth. When I started, I said to the neurologist, we can test only type 1 CMT, so this is many years not more valid. But from the devil is in the detail. This 127 genes, 120 or almost 120 are extremely rare. That's what was shown also in the workshop what I attended about the therapy. The crucial genes which can explain 90 or 95% of clarified patients are only about 8 or 10. I think this is important to keep in mind that next generation sequencing did not replace the classical methods. It's important addition, but for example, from our experience in Prague, when we clarified over 2,000 or 2,200 patients in this 25 years, but when I looked 2 or 3 years ago, how many patients needed next generation sequencing, it was only about 5%. So 95% were clarified because we were able to recognize approximately the type. So I want to stress that it's important to investigate the patients by experienced physicians in the centers. What's important that something like is now available in Europe, these rare diseases centers, that the more frequent type are usually well recognizable by experts, and it's good to use a rational approach, not to do exome in every patient, but try to get the clinical data, the family history, and work with that in the indication for genetic testing. When I started, then first the patients were interested for the genetic diagnosis because, for example, prenatal diagnostics. But when we were able to clarify their cause of the disease, they asked for therapies. This is clear. So therefore, we organized a group from orthopedics and physiotherapists to help them, but is also available only for part of the patients. So therefore, I'm happy that now I see there are also therapeutic approaches, and I'm also now recently involved, first time in my life, in clinical study in SORT patients in Prague. And I am confident that the therapy will be available and probably soon, because it's already available in other diseases. What we see the big successes in SMA or even hereditary neuropathy as amyloid neuropathy. This is curable or at least disease which is possible to stop. And there are methods available for treating other diseases. So I'm confident that this will be able to use also for Charcot-Marie-Tooth. So thank you.
So thank you very much, Pavel. So I think we should open this to discussion. As far as I can see, 4 themes have arisen. 1, sharing data. 2, accurate diagnosis, and particularly the problem with variants of unknown significance. 3, trial readiness, and how we get our therapies to where they need to be, that is into the nerve or Schwann cells. And 4, how we manage patients right now with therapies and surgeries. So this is open to any questions, any comments to anyone that's spoken. Peter, and I think we should give a round of applause to Vincent and Peter, who found the first gene for CMT. I think to have them here is amazing.
What I would raise is that I've seen it from the beginning to now, and I've been involved in other projects also. But what I see recently is that there is a lot of shift in funding, for instance, in a sense that it's more top-down now. And I wonder, in Belgium, for instance, when you do something with patients and WES, your projects are just cancelled because it's medicine. And now I hear about collaborations and registers and so on. But I see a tendency that the money goes to the top and then it trickles down and there ends the work, but there is no funding at the base. That's my impression. That's what I hear from my younger colleagues. We have to do the work. We have to fill in the registries. We have to follow our patients with novel scales and so on. It's a lot of work. And then the funding agencies tend to say, yeah, but that's in your clinical files. And to some extent it is, but it's not. I mean, there is a difference between seeing clinical patients and doing kind of clinical research. We don't have the work for that. I've been involved in epilepsy, for instance. It's like 20,000 patients are now studied in Broad Institute. They pay for it, but it kills the local groups. You don't get any funding when you say, I'm going to study 200 patients. Say, but you're involved in the Broad Institute, 25,000. Why would we give money for you? And this is a tendency I see, and I think we have to resolve that.
So Peter, do you think it's, I mean, I think that's an incredibly important point.
And may I make another point? It's like two other points. I've been invited once, but that was for epilepsy also, with people of NIH. And they had a big conference there, and they say, look, we are lagging behind in the molecular genetics of epilepsy, and we want to catch up. Give your ideas. What kind of projects would you like us to fund? But for instance, say, GWAS, we are not going to fund. We are not interested. So it's not like you have to apply for, with your own ideas. They say, we are going to steer it in some way. This is something we need. If you come up with good ideas, we will provide the money. And the second, it's idealistic. One of my former bosses went to the States and ended up with the Bill Gates Foundation. And for instance, what you see is a total different approach. What they say, we are not interested whether the results can be published, and we just indicate the things. We need a test for malaria that's usable, easy to give immediate results in hot weather. We identify the groups that can provide that, and we support them for some time. But they buy the kind of the expertise they need. I'm not going to say that's a good model for CMT, but I think like patient organizations could be helpful in saying this is the crucial information we need. Even if it's not possible to publish this as individual groups, we need that information, and to stress with the funding agencies that there is a difference between something that can be published. I'm not against publications. I have a lot of publications, a few important ones, and all the rest is noise, I would say. But I think we have to identify the things that we really need to make progress, and to make sure that the funding is coming to the people who have to do it's good to say, oh, artificial intelligence and go, but you need bioinformatics who can go through your files. I mean, in our hospital now, it's all standardized, but we have no support to extract the data. So all our trainees are putting a lot of effort in in a standardized ways to provide the data in the files, but there is no funding for extracting anything.
No, I think we could open that up, because I think one of the issues is what patients deserve now when they come to clinics is to be seen in a modern way. That is, it's often that they need proper scales done, proper scoring, proper time, but the health systems generally haven't caught up with that. So to me, it's not just funding from research, it's actually the basic funding of what is the model of care that a patient in 2023 should expect with CMT. And maybe that's much more, a political, government, health systems. I wonder whether other people have any comments on what Peter has said there. Stefan, the US, of course, it's all perfect.
Of course. Look at our elections. Peter, you brought up a good point. So we talked about this even 10 years ago. It's sort of a warning from my end, maybe even if you remember that, because I was very active in large GWAS studies back then. And it's the consortiumization, or whatever you call it. It's in more common diseases, these large consortia. What they do is they build these pyramids. Unfortunately, it just happens. And then on the top, there are like 5 analysts. They get to analyze all that work that nobody even knows where this is coming from in bringing in patients and so on and so forth. And we, for better or worse, we get in there. It's good because it tells you we're maturing, even in a rare disease. And to some degree, there's no way around it, maybe. But it does sort of, the young physician on the ground, so to say, might be not see the fruits of their work or feel kind of alienized to that big project. So one way out maybe is sharing data, like we try to do for years with this, say, Janus platform, and others do similar things. Let everyone participate in this. Just don't make it such a specialized analyst thing. Make it so that every student could technically participate in that if they want to. Because they will also find out it takes a lot of work and sweat to do the analysis. It's not just complaining, I can't see the data, it's also then putting the work in and finding stuff. So.
But I think that's a very important point. But I think there is a problem at the people that are seeing patients, bringing them in. I think that is a real issue. The other thing is, patients own their data, and I do think the only way we're going to solve data sharing is patients taking back ownership, in the sense of saying we do not think people should be doing stuff
100%.
Unless they're doing it the way we want it. And I actually think that would work.
I think so. And that's sort of to all the patients here and organizations. I think we talked about it. Don't hold back in lobbying hard. This is your data. This is your health. You would not believe it. In universities, Ingolf, you may give us blood or data under certain, that you sign a consent. Then 5 years later, I say, well, but there's this new thing, we want to do this with this blood. And they say, you can't do this. He didn't sign for this. So I can't. It's crazy. So that happens all the time. Yeah. So you need to take in your hand more. That would be
Angelo?
No, I agree with everything, of course. The problem is also, we had, this talk with our association, the knowledge of the disease at the general practitioner level, which is basically, lacking, I think. And actually, I don't know if Filippo is here, they are starting a program. Oh, okay. They are starting a very nice program, a platform for, giving to the general researcher a better knowledge of the key aspects of the disease, because this is again a way of having the patients, and also I think involving the patient in the research.
But do you not think, so one of the things in the UK, an average GP is about 2,500 patients. So they've either got no CMT patients or a family. So I think the most powerful way is when a patient goes in, they provide the information to the GP or the website or the link. Because a GP that has 0 to 2 or 3 patients will not go to an educational forum on CMT. Absolutely. They have to have a good website or a good way of looking it up, don't they?
Absolutely. I think so, yes. I think I agree. There should be, for them, an easy way to address the patients. Exactly. Filippo, do you want to add something to this?
We've no comments from patients yet. Any? And I'm aware of time. If it's
Yeah.
a hard 6 o'clock, let me know.
Here is a comment from a patient.
Okay?
Yeah.
Not a hard 6. then.
Yeah. It's about this GP course actually, which, as you mentioned, yes, we could come to the GP with all the information, but the thing is, if we don't have a diagnosis, and especially when we're kids, then we do need to find some pediatrician and some GP who know which are the main symptoms and how to recognize them. That's why we are stressing on that aspect. And we are organizing with this course for free for general protection all across Italy. That's it.
Sounds like a very good idea. Tanya.
So with scales. And we were dealing with that with SMA. In France, 3 years ago, we had two drugs for the adult patients. And when we had a committee with the government or the health, they said, okay, it's very expensive, but go on. And how will we evaluate the patients? You can manage to find some physiotherapists, but we don't have money to pay these physiotherapists. We don't have enough physiotherapists. You have to manage ourselves. So we have some funding from IFM to manage to evaluate our patient, because how you can say that a drug is working if you cannot evaluate your patient? It's not just saying, I'm doing well. You have to have skills. And that's really the problem with CMT. If we have a drug, we will have to be evaluated, and we will face the same problem. The physiotherapies or scales have been done. And I thought about something. We have 3 pharmaceutical groups in SMA. I think the government should say a percentage has to be made to provide physiotherapists to the scales. It was the same thing for multiple sclerosis. When multiple sclerosis came with the treatment, all says we need MRI to evaluate patients. And they've had MRI, the government and the labs. So we have to make some pressure, to the government, the pharmaceutical group, to say we need some physio, we need some, additional doctors to evaluate the patient, because it's time consuming.
I mean, I think that's an extremely good point. I've seen it in TTR amyloidosis. I am astonished that we can prescribe drugs £300,000 a year, but we're not given anything extra to follow them up, or to actually, I'm absolutely astonished that anybody would let us do that, but they do. Mikhail?
Peter's very well taken. I mean, in clinical practice, we see the same thing, right? That we're trying to establish the nice scales we have, but it's not very rewarding if assistant doctors don't really have the feeling that they're part of the whole funding process, and in the end just do their job and then move on to the next rotation. And I'm wondering, maybe the question to the organizers here of the CMT Federation. I mean, we want to get there where the INC is, basically, right? I mean, if we think about, I think it's the 4th funding period or the 5th even, the INC has gone through, with 4 years each, right? So in Germany, you get 3 years and that's it, right? You're lucky to get a second one. And now how to build up an let's say, long-term a network established, and all the beautiful things we'd like to do for our patients needs a very strong patient organization. And my hope is, I could just maybe put this back to the organizers here, is that the European CMT Federation has such a strong voice, just like in the US, probably the patient organizations, of which some are here, have probably vocalized their sorrows very loudly, and then NIH was giving them money. Now, and it really brings me to the point that you brought up, Mary, and that is fragmentation. Now, if you're confronted with 20 different organizations, and we saw some of them up on the wall, I mean, we should think about, really, as a result of this meeting, to use or maybe apply the established, well, the CMT Federation, but also the CMTRs possibly, in order for us to speak with one voice and give the funding issue, which we're all talking about, some more weight. And another thing which goes along with that is the question of, I don't know if this was discussed in the second part, in the second workshop, and that's the question of IIT studies. So some of you may know we have drugs, or we have ways of therapy that we could apply, which of course no pharmaceutical company is going to push because there's no money there. So what we could do is an IIT study. I just want to bring that as an additional point, and just...
That's a very important point. Very good points, actually. And I think there's a lot of commonalities coming out with the points here. You do realise you just seem to be getting jobs and jobs and jobs. You may not have felt it was coming this way, but this, I think, reflects the enthusiasm for people to work together with patients. And I actually think, we are, privileged to look after patients with this condition, but perhaps the partnership needed to develop more in many ways. And okay. Well, they are, and I think they're a nightmare. I have one starting next week that has, I can't tell you what the last 2 years has been like. They're a nightmare in terms of getting them through all of the various, things you have to get them through. And that's something we certainly could all do with help with. I know we're time-wise running a little bit out, but I just wondered if there was any comments that anybody wanted to make. I thought one thing I would say, I was thinking coming over this morning, I was, in the Eurostar, and I was thinking, as you do very early in the morning, that 200 years ago, if somebody said you could go underneath the sea from England to France to get to a meeting, you would probably have been burnt to the stake. So the only thing that limits what we can do is the lack of imagination. Any final comments from the two organisers? Oh. Or do you just want us to say the bus is coming at 7:30, is that right, tonight? 7 p.m. Bus is at 7 p.m. in front of the hotel. Okay. 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 Thank you very much to everyone. Thank you.